Good morning, everyone, and welcome to the Garvan Institute of Medical Research. It's so lovely to see many familiar faces in the Garvan auditorium. Thank you so much for coming in person. It's really lovely to be able to connect face to face in this new normal world that we're living in. We also have, I believe, more than 100 individuals in the Zoom room. So welcome to all of you joining us online as well. It's just a delight to have you. For those of you who don't know me, my name is Tilley, and I have the great privilege of heading up the Garvan Research Foundation, which is the fundraising and marketing arm of the Garvan Institute of Medical Research. I've been lucky enough to be here for just over 12 years, and it's been an amazing 12 years, and I couldn't be more excited for the future. And today's topic is something that's particularly close to my heart, and no doubt it is for many of you here as well, and those of you in the Zoom room. Before we commence, it's my great honour to acknowledge the traditional owners and custodians of the lands on which we meet, the Gadigal of the Eora Nation, and I pay my respects to elders past, present and emerging, and acknowledge their ongoing connection to land, waters and culture. Wow, pancreatic cancer. In the 12 years that I've been here, I've witnessed firsthand a phenomenal transformation in our understanding of the underlying cause of this disease, the complexity of this disease. And today you're going to hear from two incredibly talented scientists on the work that they're doing to help not only improve outcomes for individuals with pancreatic cancer, but a very bold vision to triple survival rates by 2030. 12 years ago, the average survival for pancreatic cancer was only 7% over 5 years. With thanks to efforts of the Garvan team and teams around the world, that has now increased to 12.2%. I know that doesn't sound like much, but that is a considerable step change. During the last 10 years, there's been some really important work that's been led to understand the underlying complexity of pancreatic cancer. And part of that effort was led out of here by Garvan with the Australian Pancreatic Cancer Genome Initiative, part of an international effort to characterise the molecular basis of pancreatic cancer. And in doing that, we and others discovered that this is one of the most heterogeneous diseases that there is, and that is in part why it's been so difficult to improve those outcomes. Another issue has been the late diagnosis of pancreatic cancer, which I'm sure the team will further explain soon. So we've got two of our young superstars. It's always good to have some diversity. We have a lot of people come through the public seminar series. We've got Dr. Sean Porazinski, who is a group leader within the within the personalised cancer therapeutics team. And we have Dr. Kendelle Murphy representing the personalised cancer medicine theme. Guys, thank you so much for being here. In terms of format, we're going to hear from Sean and Kendelle, and then we're going to open up for a Q&A. We will be welcoming questions online and from those of you here in the room. For those of you in the room who would like to ask a question, just pop your hand up and the team will bring around a handheld mic. And for those of you in the Zoom room, please do enter those questions at your leisure, and I'll do my very best to get to all of them. Without further ado, welcome, Sean. Thanks.
So just checking everyone can hear me okay? Up at the back? Yeah, great. Okay, so thanks to Mara-Jean for the really nice introduction to today's kind of plan of action. And I'd also like to thank the Garvan Foundation for putting this event on and giving us the chance to showcase our work here. So as Mara's kind of alluded to, I'm going to be talking about our precision medicine program in pancreatic cancer. And I guess many of you in the room are already fairly aware of what a dismal disease this is and how bad the prognosis is. But for those perhaps who aren't, or just to put some numbers behind those claims, at the moment, it's estimated to be the third leading cause of cancer deaths in Australia. But by 2030, if we don't reverse the current trends, it's going to be the second. And at the moment, there's very little in terms of curative therapy. So the only real option is surgery. But unfortunately, most patients present with advanced disease and are not eligible for that surgery. So they tend to get chemotherapy, and even the two best chemotherapy combinations that we have only improve survival by months. So it's really not good enough what the current kind of landscape for treatment. So we're really kind of dedicated and passionate about changing that. And as Mara-Jean mentioned, the 5-year survival rate has improved, but it's still really terrible at around 11 or 12%. So we think we can do much better with that. So I guess, as I've just kind of started alluding to, one of the kind of major clinical challenges with pancreatic cancer is this therapeutic resistance. So most patients present with metastatic disease. So that's disease that's already spread to other parts of the body. And as Mara mentioned, our kind of previous efforts within our lab, but also with teams around the world, have really shown how complex this disease is. So no two patients are the same. And therefore, this kind of one drug fits all approach is not going to get the job done. So we're really, committed and focused on developing these more tailored or personalized treatments that are individual to the patient. And what I want you to bear in mind for the kind of rest of the talk today is as we know more about this disease and we delve further into the mechanisms driving it, we're really understanding that pancreatic cancer, as well as other cancers, exist as ecosystems, okay? So the bulk of the tumor is made up of these cancer cells, but you have all of these supporting actors around the cancer cells or around the tumor that are playing a role in promoting the aggressiveness and progression of the disease. So you can see that in these illustrations here. So the blue cells kind of represent the cancer cells, and then you've got these other cell types shown in the other colors around the cancer cells. And all of these cells talk to each other and really kind of work together in unison to drive the disease. And what's really important is that we know, we're starting to know more about these kind of ecosystems in the primary site, so in the pancreas. But because so many patients already present with disease that's spread, we also have to make sure our treatments are targeting these ecosystems in the distant sites, okay? And so just to also highlight another key feature of this kind of cancer ecosystem, there's also a component that's non-cellular. And this is this kind of dense meshwork of sticky proteins that encapsulates the tumor and really prevents us being able to get drugs into it very well, and also allows it to hide from the immune system. And so I'm going to talk about that more in the first half of my talk. So I guess, as Mara-Jean's already alluded to, we really have some lofty ambitions in the personalized cancer therapeutics lab. And we're really trying to position ourselves to reshape outcomes for patients diagnosed with pancreatic cancer. So how are we doing that? Well, we've established this really kind of in-depth program that is bi-directional. And what I mean by that is that it goes from the bench, so discoveries in the lab, and then we take those into the clinic, which we call the bedside, and then we take those clinical findings and use those to go back into the lab and then inform the next generation of treatments. So this is a really busy slide, but I don't want you to worry too much about the details. It's just simply to highlight the breadth and depth of our program. And so we take our kind of candidate treatments that we identify in the lab. So this is shown on the left. So we have a wealth of beautiful models here, an infrastructure here that allows us to really investigate the effectiveness of our new drugs or our new combination treatments before we go into clinical trials. And a lot of that activity is happening with the MOST National Clinical Trials Program, which is run by David Thomas. So just to give you a quick overview of the talk today. So in the first half, I'll be talking about a project targeting this meshwork, or this fibrosis, as we call it, that encapsulates the tumor. And then in the second half of the talk, I'll be covering a really interesting project that is looking at repurposing an antifungal in pancreatic cancer. So the first half of the talk, the work has really been kind of spearheaded by a very talented PhD student in the lab called Ben. And he's been taking a drug that we call the porcupine inhibitor. And what this drug does is to target the pathways that lead to the generation of this fibrosis or meshwork around the tumor. Okay, so I mentioned that there are lots of different cell types in the cancer ecosystem or in the tumor ecosystem. And one of these key cell types is called fibroblast. And you can see it there on the diagram shown as the kind of long blue thin cell. And what these cell types do, they're the guys who produce all of this dense protein meshwork that encapsulates the tumor. And they're very motile cell type. They like to move around on this meshwork, and they like to remodel it and squeeze it and restructure it and reorganize it. And what this does is it allows ultimately the cancer cells in the primary site to then start migrating and invading out of the primary site and spreading to distant sites. So this is just a really kind of simple experiment that we do in the lab, where we take these fibroblasts, we embed them into this kind of a mimic of this meshwork. So in this case, it's collagen. So not the collagen you might have, had into your cheeks or your forehead to improve your wrinkles, but it's one of the main constituents of this dense meshwork in the tumor. So what we can do here is kind of mimic how our drugs affect this meshwork in the plate in the lab. And so all I want you to kind of take home from this is that our drugs, so the porcupine inhibitor, which is shown in the kind of bottom row of wells, stops the collagen disc from becoming smaller. And so we know as these fibroblasts remodel the collagen, they decrease the size, and you can see that in the top row in the untreated wells. But when you look at the bottom row, you can see that the area remains larger when we treat. So that already tells us we're blocking the activity of this cell line. And then these are just the kind of quantifications of that below, but I think it's nicely represented in the images. And so that's all done in the dish, which is great. But here, I just want to show you that we can also look at this process in vivo. So this is just a movie of us. If I can get, yeah, here we go. So this is just a movie of a tumor in the mice live. And what you can see here is, again, this meshwork is shown in purple, so the collagen. And then you'll see that we've taken a scan down through the tumor. And what you'll see is that the cancer cells are shown in green, and then the blood vessels are shown in red. So I just want to highlight the power of this approach to be able to visualize things in real time, in 3D in the mice. So it gives us a really good idea of how our drugs working in the context of an intact tumor. Okay, and so then what we have here are just stills, 3D stills from these movies. And what you can see on the left is the untreated tumors. So there's still that real presence of the collagen or the meshwork. And when we look on the right in the porcupine treated tumors, we see a real reduction in this. And this is exciting for us because it tells us that we may now be improving our ability to deliver drugs into the tumor. Okay, and then in kind of support of that, what we also know is that these tumors are this meshwork is very dense and tends to constrict and compress the blood vessels within the tumors. And so what you're seeing in the top movie, which I'll just try and play again, so we can inject these red fluorescent dots into the tail vein of the mice, and then watch how these dots travel through the bloodstream and how they are able to kind of get into the tumor and dissipate throughout the tumor. And so if you look on the left, what you can see is that the dots are mainly contained within the blood vessels. So this is an untreated mouse. So again, that meshwork is compressing the tumors and stopping the ability of any drugs to perfuse out of the blood vessel. But when we look at the right-hand treated tumor, you can see that there are lots of dots now outside the blood vessels. So that really tells us that the permeability of these blood vessels has been improved. And then in the bottom, we've just got 3D, again, 3D representations of these movies. And hopefully what you can appreciate is in the right-hand movie with the treated, there's a lot more of the fuzzy red outside the blood vessels. Okay, and so the other thing we're now doing is start to kind of take a more kind of holistic look at what our treatments are doing in the tumors. So as Mara-Jean alluded to, that there is a lot of molecular complexity with pancreatic cancer. So no two patients are the same, but we also know that different areas within a patient's tumor are different. So we really want to understand how our treatments are working on a kind of global tumor scale to affect these different populations. And so we're using this approach called spatial biology now, which allows us to see what individual cells are doing in different parts of the tumor and how they're responding to our treatments. And so this is just some example of the work we've been doing in this space. And so again, this is quite a detailed slide, but the kind of main message is that we can first of all stain the tumor tissue with different markers that will highlight the different cell populations. So on the left, you've got the tumor cells, they're stained in green. And then on the right, you've got those fibroblasts that I was talking about a couple of slides ago, and they're shown in the red and green. And what's really nice is that we can sample the areas as highlighted by the white circles, and then look at the gene expression or what's going on at the molecular level in those cells in the different areas. And what's really nice is that if we look at the right-hand grid, so this is just a kind of a readout of the experiment that shows us what's going on at the gene expression level. And if you just focus on the left half of the grid, which shows the tumor compartment, what you can see is that there's a big difference between tumors that are not resistant to chemotherapy and tumors that are resistant to chemotherapy, shown by the kind of purple box. And so the color scale highlights whether the gene is on or off. So blue is off and red is on. And what you can see is that in the resistant tumors, so the kind of last three columns of the left-hand part of the grid, you can see that there are a bunch of genes being switched on in response to the chemotherapy driving resistance. And these genes are all kind of involved in this remodeling of the meshwork and in the kind of migration and metastasis of the cells. And what we've now been able to do is take the porcupine inhibitor and treat these tumors that have become resistant to chemotherapy, and we've actually been able to reverse that resistance. So that's really exciting. Okay, so for the second half of the talk, I'm going to switch gears a little bit and talk about quite an interesting project for us in terms of repurposing an antifungal. So this is, again, a project that's now been heavily worked on by Jen Mann, who's a PhD student, and also one of our senior computational biologists, Diego. So this project really started off the back of a super interesting case study in the literature, whereby a patient with advanced disease was placed onto palliative care, and then after several months developed a lung infection. And so they stopped all of their chemotherapy and just started taking this antifungal called itraconazole. And what's kind of unbelievable is that 9 months later, they were still alive. So we know that this just doesn't happen in pancreatic cancer. So had that person stayed on chemo, there's no way they should have been alive. But just to be on this antifungal alone and still alive 9 months later was incredible to us. But what's even more outstanding is that when the patient went back into the clinic, their disease was now so regressed, so shrunk, that it was actually surgically operable. So they underwent a Whipple surgery and they were still alive 4 years post-surgery. So this is just, to us, kind of unbelievable working in this field. And so what's really nice about this drug is that it's FDA approved. It's already been given to tens of millions of patients worldwide. So we know it's really safe. We know how well it works. We know how to administer it. And because of the changes that we saw in this kind of exceptional responder patient, it suggested to us that the drug was doing something at a whole body scale. And to us, that initially kind of jumped out as being, maybe it's doing something with the immune system. So we wanted to ask the question, can itraconazole make the immune system better at detecting and killing pancreatic tumors? So just to give you a kind of brief introduction or overview of the immune landscape in pancreatic cancer, and this is true of many other cancers. So you initially have these kind of cancer cells arising in the body, and the job of the immune system is to detect them and kill them, right, and get rid of them. And so if you look at the schematic here on the left, you can see all the different immune cells shown in the different colors. So they all work together with the common goal of eliminating these foreign invaders or these foreign objects. But what you can have, and this is what we call the equilibrium phase, you can have tumor cells that arise with some kind of advantage or some mutation that allows them to coexist with the immune system. So the immune cells can see that they're there, but they're not really able to kill them. And so they kind of coexist in harmony. But then as those kind of tumor cells expand and grow and proliferate and divide, and the disease progresses, you eventually get a full shutdown of the immune system. So now we term this the escape phase, and now you've got a situation where the immune cells can no longer detect or kill the tumor cells, and we get uncontrolled tumor growth. So we're really interested in asking whether itraconazole can restore the balance here. So we've started to do some experiments at the kind of late phase, the escape phase. So this is the phase at which most patients present with clinically. And so what we've been doing is looking at a variety of immune cells within our tumors. And so what you can see shown in these kind of circles on the right, so the immune cells are stained brown. And you can see that this top, in the top panel, this is what we consider a bad immune cell. So the role here for this immune cell is to really dampen the activity of other immune cells in the environment. And what's really nice is we can block these bad guys when we treat with itraconazole. So we see a reduction in the number of these cells. And then when we look at these cells at the bottom, so this is a cell type that's involved in actually performing the killing of the tumor cells. And what we see there when we treat with itraconazole is an increase in that immune cell type. So we really think that we're kind of switching the immune landscape to be one where it's going to be more anti-tumor. So these analyses are great, but they are static. And we know that immune cell interactions are really fast and dynamic. And so, together with Paul Timpson and the ACRF site Insight Center, we're now planning to start work where we are going to actually look at how itraconazole is affecting our immune populations in real time in the tumors. So this is just a video that shows our kind of capabilities. And what you've got here again is that dense meshwork, or the fibrosis shown in purple, so the collagen. But you can see that the immune cells are labeled green. And so what this will really allow us to do is to see how our immune cells are behaving following treatment with itraconazole. And so because of these nice immune changes we're seeing in the treated tumors, we decided to combine the antifungal with immunotherapy. So classically, immunotherapy has not been good in pancreatic cancer because there are so few immune cells in there, and the ones that are in there are not doing their job. So we kind of felt that because of this nice reversion of the immune landscape that we were seeing with the antifungal, it made sense to combine with immunotherapy. So what you've got here at the bottom is what we call a survival curve. And so again, you don't need to worry too much about the details, but the kind of key point to focus on is where the lines meet the time axes. And so each different colored line represents a different treatment group. And you can see that if we have untreated mice, they succumb to, or all the mice have succumbed to ethical endpoints by around 40 days. And you can see immunotherapy doesn't really offer any advantages, which we know from the clinic. Then we can see that itraconazole extends the kind of survival time to just over 60 days. But what's absolutely remarkable is that if we combine the antifungal with immunotherapy, you can see that at 80 days, all the mice are still alive. So we're really excited about this. And this is our next kind of best candidate for a clinical trial that we're really hoping to get started within the next sort of year. Okay, and so just to finish with some of the newest stuff we're doing in that space to understand the kind of ecosystem and the changes that our treatments are causing. We're now using this technology called single cell sequencing. And what this allows us to do is to take these tumors, break them up into their kind of individual cell components. And then what we're able to do using this technology is look at which genes are on and off within individual cells from the tumor. So this is now really giving us an unprecedented resolution into and wealth of information into what's happening in our tumors in following treatment. So we then take this kind of single cell soup, as I like to call it, and then we can sort all of the different cells into their kind of constituent compartments. So we'll take all of the tumor cells into one tube, we'll take the fibroblasts into another tube, the immune cells into another tube. And then we run them through these microfluidic devices. And what happens here is individual cells are encapsulated within an oil droplet. They're broken open to release all of their genetic material. and then we can start to sequence that genetic material and look which genes are on and off in which cells following treatment. And so this is how that how that data looks, kind of unspectacular considering the complexity of the experiment. But what you can see here is what we call a kind of UMAP plot. And what this does is groups all of the different cells together based on their gene expression. So you can see that each single dot on the plot represents an individual cell. And you can see that all of the similar cell types cluster together, which we would expect, right? Because they're the same cell type. They should have similar gene expression profiles. But what we can then do from that is start to look deeply into some of these populations. So this is one of the immune populations I mentioned a few slides back. So these are the macrophages, and they in pancreatic cancer, they exist to kind of really suppress the other immune cells in the environment. And so there are kind of two types. There's the type we call M1 and the type that's M2, and that's bad. And basically all I just wanted to illustrate here is that you can see when we treat with our antifungal, we cause a switch in the proportion of these cell types. So that's shown in this bottom right bar graph. So you can see that in the untreated state, most of these macrophages are in the M2 state. So this is the bad state. But when we treat with the antifungal, we see a switch back towards this M1 state, or this good state. So that has anti-tumor properties. So we're really able to now see at single cell resolution how our treatments are kind of reprogramming the cells. And so I'd just like to finish there by thanking everyone in the team, as well as all of our collaborators, our funders, past and present, as well as all the patients and families who've donated their time and support and material to the projects I've talked about today.
All right, just checking you can hear me out the back. Yep, all good. All right, so luckily Sean's kind of introduced half of what I'm going to talk about today. But pancreatic cancer, as Sean's already mentioned, is quite a complex disease, which involves not only the tumor cells, but also the structure and the architecture that surrounds it and supports it. And this we call fibrosis, or ECM. So as the disease progresses from a normal pancreas through its different stages to a full-blown pancreatic cancer, this fibrosis increases throughout the disease development, and it's thought to be a barrier to treatment, so it prevents chemotherapies and other different treatments getting into that tumor. But it's also thought to promote disease development and also to help it spread from the initial primary tumor to secondary sites. Now, if we take some samples from a fibrotic cohort of patient tumors, you can see here that in a normal pancreatic cancer, there's quite a bit of fibrosis, which is highlighted in sort of the dark pink/red. However, when we look at patient tumors which have been treated with chemotherapy, you can see there's a really big increase in the amount of this fibrosis that is occurring within these tumors. So the chemotherapy is causing a sort of a wounding or a scarring, creating more fibrosis, which then acts as a further barrier to the next round of chemotherapy. And this just keeps cycling and cycling and cycling until there's so much collagen that the chemotherapy can no longer get into these tumors. So what we want to do is to try and target this fibrosis. Can we sort of change it a little bit, reduce its amount and its structure to try and allow chemotherapy to actually get into these tumors and do what it should? So in order to do this, we're targeting a protein called focal adhesion kinase, or FAK, which is at the barrier or the edge of cells. And it's responsible for talking to this fibrosis and also from sending messages from this fibrosis into the cancer cells. So you can see here, there's sort of a line between the fibrosis and the cancer cell. And we want to see what happens if we can sort of stop this talk. If we can cut off this communication, can we slowly reduce the amount of fibrosis that's occurring in these tumors and improve subsequent chemotherapy treatments? Now, in order to do this, we look at our cancer in a sort of personalized manner, which Sean's already highlighted. So we take tumors, patient tumors from the Australian Pancreatic Genome Initiative. We have a look at the amount of fibrosis in these tumors, and then we also look at the levels of FAK, which is our target in these tumors. And what we can see is that if we target, or if we try and target FAK in a proportion of patients, we can potentially improve their survival. So for example, if we look at this graph here, in patients with a low amount of fibrosis but high levels of our target, it's actually prognostic of a worse survival as opposed to those with a lower amount of our target, which suggests that if we do target this, we could potentially improve survival. So in order to do this, we take patient samples to see if we can have a look at how different patients respond, because as Sean's already highlighted, every patient's tumor is different, and also within these tumors, there's fine intricacies as well. So just for this talk, I'm going to focus on two particular patient samples, one here that has low FAK, so very little that we can target with our drug, or high FAK, which has high amounts of FAK that we can therapeutically target. So in order to do this, we use models of the disease where we inject cancer cells and then treat these tumors with our FAK inhibitor prior to chemotherapy, so to reduce that fibrosis before we even treat with chemotherapy. So for example, we have a 3-day treatment of our FAK inhibitor, a little break, and then chemotherapy. Now, what we can see here is in our insensitive or low FAK cell line, we see no real response to treatment between yellow, which is our chemotherapy alone, or blue, which has received this pre-FAK treatment. However, if we take a patient cell line that has lots of this FAK and then treat with either chemotherapy in the yellow here or with our FAK inhibitors prior to chemotherapy, what we can see here is there's a marked increase in the survival, which suggests that we're rendering these cancer cells quite sensitive to subsequent chemotherapy. Now, a little bit of magic going on here. We need to work out a way to look at which patients will and won't respond to this treatment. We've already seen that there's one cell line that has low FAK and doesn't particularly respond, and one that has high and does respond quite well. But is there a second marker that we can use? And something quite useful when we're doing research is looking beyond the cancer that we're actually working in. So obviously we're working in pancreatic cancer, but if we look at, say, mesothelioma, for example, or asbestosis, people have seen that there's a relationship between a protein called myelin and sensitivity to FAK inhibitors, such as the one that we use. So we then looked at our two different cell lines, and what we saw was that our insensitive cell line had high levels of this protein called myelin, and our high FAK cell line, which was super sensitive to our FAK inhibitors, had low levels of myelin. And these sort of different predictions can allow us to then tailor treatment in a personalized manner. So we wanted to just double check and verify that this prediction was actually true. So we took our previously insensitive patient sample and we did a bit of scientific magic and we completely removed this protein called myelin. So it's completely gone from tumors and they should in theory now be sensitive to our FAK inhibitors. So you can see here we have a tumor treated with chemotherapy only, and one here which was pre-treated with our FAK inhibitors prior to chemotherapy, and there's no real difference in this particular setup as we expected. However, when we've removed this protein called myelin and we do our FAK pre-treatment prior to chemotherapy, and I know this graph is mildly confusing, I got confused at the first time too. What we can see here is instead of the tumor being the same size as chemotherapy only, it's now actually about half the size. And when we look at the mildly confusing graph, I want you to focus just on this dotted green line at the bottom, where we've actually managed to stabilize disease. So this is just highlighting that sometimes there's more than one signature or one marker which can predict a patient's response to a particular therapy, and that we should go in and maybe have a look at how different each tumor is and what markers will actually be useful in stratifying treatments and giving therapeutic options within a clinical setting. And this just highlights further that there really is this sort of subpopulation of patients that we can target, and hopefully, even if it's only a small percentage, we can move that 12% 5-year survival rate to 15 or 20%, and slowly, patient by patient, we can increase that. Now, how do we do this? And Sean's already shown you a bunch of beautiful images that we take down the microscope. So why are we using imaging to actually look at our tumors? We can take static endpoints, but that doesn't really tell you what's actually happening in a live tumor. It's a fixed sample. So we use the microscopes to actually look what's happening in real time, when and where our drugs actually working, and when are they targeting cancer cells and when are they not. So in order to do this, we look at the tumors in whole using some of the really powerful microscopes that we have at the ACRF Insight Center. And what you can see here is a whole tumor. And the tumor cells here are in red and green. And you can see that they're sort of encapsulating themselves in that white fibrosis. So we can now watch and see how these cells not only respond to our drug, but also how they remodel this fibrosis in real time. And so we use different traffic light systems, shall we call them, to be able to see if our therapies are working, when and where. So for example, at the top here, you can see there's no treatment here. Cancer cells are active, they're nice and red. However, we've given some of these cancer cells here some chemotherapy, and you can see they've actually switched to green, which to us indicates that our chemotherapy is actually working in this region of the tumor. We can also use a different traffic light system, and we call these biosensors, and these are usually tagged to a specific protein. So for example, these ones are tagged to FAK. And what we can do is, again, we can see that color change and see here in our non-responder line, the cancer cells are all sort of this yellowy, greeny color, and when we add our treatments in, they don't particularly change. However, in a responder line, you can see here that we've got high active amounts of protein, as indicated by sort of the yellow/green color. But when we add in our drugs, you can see they completely respond and switch to this blue off color. So this just lets us look at the dynamic activity of proteins in a live tumor in distance to the vasculature, for example, or different areas of the tumor. And let's just really see when and where our drugs are working. And again, we can look, as Sean's already shown you today, at the vasculature. Are our drugs changing the vasculature? Are they becoming more leaky? Are the vessels structured differently? How is this helping the chemotherapy actually penetrate deeply into these tumors? Whoops, sorry. Didn't mean to do that. Anyway, we then moved on and wanted to see how our FAK treatment regimes were actually acting in a whole live tumor setting. So again, we took the same treatment that I showed before, where we pre-treated our tumors with our FAK inhibitors, gave the mice a small holiday, treated them with chemotherapy, and then we performed live imaging using a red-green traffic light, which allowed us to see if our chemotherapy was working in these tumors. But what we also were able to look at the same time wasn't just the cancer cells, but we could look at this fibrosis. And what you can see here in the chemotherapy treatment alone, as I showed at the start, there's a vast amount of fibrosis within these tumors, which will make further chemotherapy harder to actually get into these tumors. But when we've pre-treated with our FAK inhibitors, it's had an anti-fibrotic effect, and it's reduced the vast amounts of collagen and fibrosis in these tumors, which when we then add our traffic light system in and have a look at how effectively chemotherapy is working, what we can see is that in our chemotherapy-only tumors, highlighted by the green cells, some of them are receiving chemotherapy, some of them aren't. So some of them are dying from chemotherapy, but some are still growing. However, when we've removed this fibrosis by pre-treating with our FAK inhibitors, you can see here in the proportion of green cells that pretty much all of the cancer cells are now receiving and responding to chemotherapy. So just by simply reducing the amount of fibrosis in there, we've been able to actually penetrate deep into these tumors and to actually start to see real differences with our chemotherapy. Now, this also holds true, and I apologize for the slightly disgusting image of the livers, but as Sean's mentioned, pancreatic cancer isn't only a primary tumor. One of the reasons the survival is so dismal is due to the large amount of metastases that we do see or colonization at secondary sites such as the liver. And if we take this same treatment, pre-treat our mice with our FAK or antifibrotic treatments prior to chemotherapy, you can see here in a live metastasis, so this is a small tumor that's grown in the liver, originating from the original primary tumor. What you can see here is that whilst it is responding to chemotherapy, as shown by the green cells, it's still quite large and growing. However, when we've pre-treated, we've not only reduced the number of small tumors or secondary tumors growing in the liver, we've also reduced their size and also their susceptibility to chemotherapy. You can see here almost all of the cancer cells are now green and responding to that chemotherapy. Now, what does that mean for a clinical setting? How can we actually translate this into the clinic and treat real patients? So we've started collaborating with a biopharmaceutical company in Melbourne called Amplia Therapeutics, and they have an FAK inhibitor, which we can actually use within a clinical setting. And so using our traffic light system, again, here you can see using their drug, we have high amounts of active FAK. When we've treated them, the cancer cells switch to blue, and you can see that we've turned this off. And what's more is that when we look at this fibrosis, you can see here again in a control setting, there's vast amounts of collagen throughout the tumor. And when we've treated with the Amplia FAK inhibitor, you can see that this is pretty much gone. And it's small amounts of fibrosis, which means that when we then add in chemotherapy, can we change it? And so we use the same treatment that we've been using a few times, where we pre-treat with the Amplia FAK inhibitor prior to chemotherapy. Now, in this particular setting, we're using gemcitabine and Abraxane, which is currently the standard of care in Australia. And what you can see here is that our FAK inhibitors alone, as well as chemotherapy, is actually affecting survival and increasing that survival in our mice. But when we've pre-treated these tumors with our FAK inhibitors, reduce that fibrosis, reduce the spread of cancer cells to the liver, you can see this massive increase in survival here with this pre-treatment regime. Now, is this applicable to only gemcitabine and Abraxane, or are some of the new and emerging chemotherapy treatments that are observed in the clinic also going to respond to our pre-treatment regimes? So FOLFIRINOX is a new chemotherapy which involves 4 different treatments, and it's available for relatively fit patients. So we just wanted to see if the same fibrotic reduction was applicable to this particular chemotherapy regime. So again, we pre-treated our mice with our FAK inhibitors prior to them receiving FOLFIRINOX chemotherapy. And as we saw with gemcitabine and Abraxane, there's this vast increase in survival when we've pre-treated, removed this fibrosis in the tumors, and then treated with chemotherapy. So it's just really highlighting that we need to sort of identify a population where we can fine-tune and subtly change the amount of fibrosis in these tumors to allow our chemotherapies to get in and actually do what they're meant to be doing. So Amplia has now started a phase 2 clinical trial off the back of some of this data. They're currently recruiting and have completed stage 3 recruitment of the patients on the initial dosages. So this is an early treatment where they've taken our priming regimes as a first-line therapy. Patients receive out their FAK inhibitor for a week, and then on days 1, 8, and 15, they receive chemotherapy with a priming treatment of their FAK inhibitor to try and reduce the fibrosis that chemotherapy is causing. And so we're hopeful that this will actually help some patients and benefit it as they're still actively recruiting. Not any results are available yet, but we hold out hope that we can try and change that 11-12% survival rate in the future. So with that, I have nothing to do but thank my lab, my amazing support, all our funding bodies, and everyone else involved.
Amazing. So first of all, if I can ask you guys a question that I have, how does it feel to be undertaking research that's having a direct impact in the clinic right now? You've both spoken about clinical trial, obviously the incredible discovery work that you and your teams are leading, but also the clinical trial element. And Kendelle, perhaps we can start with you.
Sometimes it's surreal. Obviously you start as a sort of scientific student going, I want to make an impact. And then you see that actually come to fruition. Sometimes you believe it, you're like, we're gonna make a difference here. And sometimes you sort of go, hang on a second, is this really happening? You have to pinch yourself. But I think it's really a team effort as well. And in the sense, you have to look at everyone else around you. And I haven't got here without the help of everyone else. And so, yeah, it does feel quite surreal, but also quite fulfilling that you are making an impact of some description. Yeah.
Sean?
Yeah, I think I would, is that on? Yeah, I think I would echo Kendall's sentiments there. So, yeah, for me, it's an honor and a privilege to be doing, for my day job, to be doing something that will make a difference to people's lives and help people. Yeah, so I always was passionate about getting into science to try and basically have, outcomes for patients. So, yeah, it's really, really humbling.
And I think working closely with patients, when you sort of see people and go, okay, if you've even given someone a couple of months or a couple of weeks or years extra that they can do what they love, spend time with their family, I think that's where it really highlights what you're doing, basically. Yeah.
Amazing. You guys are amazing. We've got some questions coming through online. And of course, if anyone in the auditorium would like to ask a question, please just pop your hand up. First one from Richard. Can you comment on KRAS gene mutations in relation to pancreatic cancer? Who wants to take that one?
Well, I guess we obviously we know they're a major driving force. That most patients, probably up to 95%, present with KRAS mutations. So there has been some strides recently where we've now. So I guess the thought in the field has been that KRAS was previously undruggable. It's a difficult protein to target. because of its size and its charge. So, you know, developing inhibitors that will bind to it and block it has been very challenging. But in the last year or so, there's been some great strides made to target particular mutated forms of KRAS. So we know that there are several kind of types that drive pancreatic cancer, but also other cancers as well. But then last week, I think there was an article in Nature, which has now shown that a development of a pan-KRAS inhibitor. So this is one that should target all mutant forms. So I think this is going to be a bit of a paradigm shift now in the field. So it's really exciting.
Amazing. Ah, there I am. I'm back. Thanks, guys. One of our long-standing supporters, Jill, is in the Zoom room. Jill says, my husband was diagnosed with pancreatic cancer about 16 years ago. At the time of diagnosis, it had metastasized into the liver. We were told that it would be the liver tumor that would kill him. He died 11 months later. Will this new research assist in the treatment of METs? And of course, Jill's husband was Dr. Stuart Furler, who was a long-standing member of Garvan's diabetes team here at the Garvan. And with thanks to Jill and the family, we do actually have an award here in memory of Stuart that we give out every year to young and outstanding scientists. Kendelle, do you want to start with that one?
I think it really, really will. We've seen, at least with our particular treatment methods, that you can not only sort of reduce how much of the cancer spreads to the liver, but you can also try and target what's already there. So once they do get to the liver, they do start protecting themselves and building this fibrosis. And if we can sort of reduce that on the cells that are already there, then we can start to see chemotherapy actually working in these metastatic sites, which we previously haven't particularly seen. So yeah, I think slowly we will start to make a difference on reducing how many actually arrive there and changing how those that are there respond to chemotherapy.
Fantastic, thank you. That's very, very reassuring. A few questions here of more of a kind of clinical nature, which is always a little bit tricky for us to respond to personal clinical questions in this context. So what I might suggest is that the individuals with those very specific personal clinical questions get in touch with the team and we'd be really happy to provide whatever guidance we can. I think I've got a hand up in the audience. Thank you, sir. We'll just get you a microphone, won't be a moment.
Yes, I'm just wondering whether you have found any connection between celiac disease and pancreatic cancer.
Funnily enough, I'm celiac myself. Good question. So far as I know, there hasn't really been any connection. It's more to do with sort of more the colorectals or the gastrointestinal cancers, where if it's left undiagnosed, there can be some triggers that do appear and increase chances of getting a gastrointestinal cancer. There's really not, that I know of at least, a really cause effect type of thing coming on. It depends how each individual manages it. In terms of pancreatic cancer, I don't believe that there's a connection, but I'm not sure. It's something I should look into.
No, why I was asking was because when I was diagnosed, I was told that, you should stick to the diet.
Yeah.
And that if you continually damage the villi, that can affect the pancreas.
Yeah.
Dramatically and cause the cancer.
Yeah, it causes, I mean, if you do not stick to the diet, it's going to cause an inflammatory response, which is going to infect not just, well, it will infect mostly the sort of the GI tract, but it will potentially expand to other organs that are in close proximity, one being potentially the pancreas. So, yeah.
Thank you.
We also, related to that, we also understand that the microbiome of the gut is important as well. It's coming to the kind of forefront in cancer research, and that changes in that microbiome can change disease progression or how people respond to treatment.
And we've got more. Oh, yes, sir. Thank you so much.
Well, thank you very much, Sean and Kendelle, for a very informative and inspiring address. And I like murder mysteries, so I feel confident that you're on the case here. My question, however, is very general, and that is, what is the relationship between pancreatic cancer and, for example, melanoma?
Hard question. I mean, look, each cancer of itself is quite individual. Some you see parallels between, for example, pancreatic cancer, mesothelioma, even sometimes ovarian. They tend to show the same treatment responses, but then you add in, say, breast cancer, and it doesn't respond how you expect. Between melanoma and pancreatic, I'd say there's less, particularly in, say, as Sean was talking about, sort of like immunotherapy. Melanoma responds insanely well to immunotherapy, but pancreatic cancer doesn't. So those two cancers, I would say, are sort of less similar than some of the others. I'm not sure if, Sean, you have anything to add.
Yeah. Yeah.
Speaking about pancreatic cancer and connections, I'm wondering, I just feel that there are possibly links between diabetes, between fungal disease and pancreatic cancer. And I was wondering if, because people who are diabetic are prone to fungal disease more, and therefore it takes me also to a prevention type mode. I had a great grandmother whose leg was amputated. Then her sons showed sugar. Her leg was amputated through diabetes. Her daughter, my great aunt, died of pancreatic cancer. And I just wonder if there are links proven in that, because I feel a very strong connection with those things, and it's gone on down through the generations.
I think we certainly know that, you know, a kind of malfunctioning pancreas in the case of diabetes is probably could be a kind of precursor or a, you know, a risk factor for pancreatic cancer. And I guess a kind of susceptibility to fungal infections would maybe suggest some sort of immune compromised kind of situation perhaps. And so, you know, as I alluded to in my talk, if you don't have that immune surveillance working well, then there's a chance that, you know, these mutated cells or these malfunctioning cells can slip through the net and then, you know, start to multiply, and that's kind of how it begins.
I think too what prompted my asking the question is the fact that antifungal treatment seems to be an important factor that you mentioned today, and it's sort of, it seems a strong link, doesn't it?
Yeah.
Thank you very much.
No? Yes. Forgive us, everyone. Yes, thank you, Rebecca.
I'm speaking for my sister who lives in the UK and was diagnosed with pancreatic cancer in November 21. She's been treated presently at Guy's Hospital, and they say the tumor is intact. She's in her late 70s. They're not going to do surgery, but she seems to be responding pretty well to chemo and radiation. In fact, she's going to Ireland today. And she had aggressive breast cancer 10 years ago, and she survived that quite well. So is there a possibility that it could be a side effect of her breast cancer?
Oh, I guess a little bit of a tricky one, because we do have to be careful not to comment on individual clinical cases. What I will say is that there certainly are correlations between some cancers, for example, breast cancer and ovarian cancer, prostate cancer in families, and breast cancer in the same families. To my knowledge, there's no correlation between breast and pancreatic. But certainly it sounds like she's been through the ringer, and I'm pleased to hear that she's responding well and off to Ireland. Probably today. How fabulous. How fabulous. Guys, did you want to extrapolate on that?
Yeah, I think I agree as far as we know. And I mean, each case is individual. Could be interesting to start looking into, but it's not something we can really add to.
Could just be, could be a common mutation that's driving, both cancer types, for example, and gave rise to the breast, and then later on has some involvement in driving pancreatic.
My father probably died with prostate cancer many, many years ago. I don't know if that has any relation. She's the only one of us so far that I know who has cancer.
Okay.
It's absolutely fascinating that the kind of the contributions that are genetic or gene based, not necessarily hereditary. Some of these genetic changes can happen during an individual's lifetime. And then, of course, there are the environmental factors. So it is always very, very complex. Sue, can we grab Sue a microphone, Eliza? Oh.
What you've just said is exactly what I was going to ask. In your brochure here, cancer is fundamentally a genetic disease. Choose your parents wisely.
Yeah, I think that goes for many other aspects of life as well, choosing you.
Well, in my family, nobody's ever had any form of cancer, as far as I know. I mean, it's interesting.
Can we join your family? Sue?
I hope my question's not too simplistic and hasn't been answered already, but is pancreatic cancer always a primary cancer, or can it be a secondary cancer?
I think that's a great question. Kendelle?
That's a good question. Usually it is a primary. I've not really heard of it as a secondary. It could be wrong, but usually it sort of starts there and then spreads at any point in time. That's the other thing with it. It will spread at any point. It doesn't have to reach stage A, B, C, or D. It will go at whatever point it wants. So I've only heard it as a primary. I've only ever heard of it as a primary, not as a secondary.
Anyone I know, it's always been a primary, but I've just thought I've never heard it as being a secondary cancer.
Not usually that I know, but then there could be, like, there was sort of that individual itraconazole case. There could be a case where it is, but in general, yeah, it's a primary.
And we've got another question in the room.
So thank you. I feel I need to disclose that I'm one of the 3 consumers on these projects. So thank you for the work that you're doing. It's really significant.
Thank you for your contribution to medical research. You're amazing.
Thank you. With the questions on hereditary factors, I'm wondering if there's going to be a presentation sometime in the future on the screening project that's being run by the Garvan. It'd be interesting maybe to get an update on that. I know that will be perhaps sometime in the future maybe, but it would be good to hear about it.
I think that's a fabulous recommendation. We will make sure that happens. And yeah, I mean, one of the most extraordinary things to be involved in medical research is just the rate of change, the rate of discovery, and the number of new genes that are being identified, and the emerging field of polygenetics or polygenomics, understanding how not just single gene drivers, but how multiple genes can, unfortunately, over time add to one's risk of developing a major disease or a major illness. I am conscious of the time. We're just past 11:30. I do know we've got a couple more questions in the audience. I'll just say, for those of you in the Zoom room, or for anyone who has a hard stop, please do feel free to sneak out. We won't hold it against you. We might just take, I think, maybe 2 more questions before we wrap up. Thanks, Rory.
Thank you for an incredible presentation. Quickly, I have 2 questions. Is there any increased incidence in siblings or progeny of a pancreatic cancer sufferer? And my second question is there any increased incidence of occurrence of pancreatic cancer from simple pancreatic cysts?
I guess related to the first question, I guess depends on the mutation that's driving the disease, I guess, and the heritability of that. But I'm not aware of kind of any hard, or I don't have any hard numbers to hand.
Yeah, I don't have any numbers either, but there is sort of some research coming up. I'm not going to name any mutations, but that there could be a connection between a hereditary gene, for example. But it's only new and emerging, so there's not really much that I can add.
And I think just to put that into context, and this data may be a little bit out of date now, but my understanding is that of all pancreatic cancers, probably less than 10%, maybe even less than 7% could be classed as strictly hereditary. So I think the message there is that if you have had a family member with the disease, don't freak. But certainly hereditary genes do play a role. I think from my understanding, the major risk factor for pancreatic cancer is actually smoking. So a good reminder that smoking is not good for anyone, for anything, and it's particularly implicated in pancreatic cancer. And I think we'll take our final question.
Excuse me. Look, I have heard that alcoholism is a common factor in the cause of pancreatic cancer. Is that true?
It runs similar to sort of smoking. So smoking and alcohol seem to have a sort of bit of connection to it. As to the specific numbers, I'm not entirely sure, but I do believe there is a small connection, yeah. Sean?
Yeah,
So it kind of, I guess it centers around that both of those things can cause, inflammation in the pancreas, and we know that this kind of inflammatory environment can be a risk factor. Yeah.
Thank you very much.
Excellent question, thank you. So everyone, I do apologise, we will wrap up here. However, if anybody has any questions that have not been answered, including some of you online with very personal questions regarding clinical trial criteria, etc., please do send those questions through to us via email, and we will do our very best to respond to you as soon as we possibly can. I think it's been a wonderful session. I want to thank all of you for joining us, both the 100+ people in the Zoom room, all of you in the audience. You are all critical members of the Garvan family. We thank you for your support, your interest, your attendance. Many of you are donors, partners for the future, corporate supporters, volunteers. We are in your debt and forever grateful for the support that you give. And may I please get a big round of applause for Kendelle and Sean. A recording of this session will be made available. Please do feel free to send that on to friends and family and anyone that you think may be interested. And we look forward to announcing the next public seminar very soon. Hopefully we'll be able to announce the second half of the year dates in July, if not before. Thanks, everyone.